Welcome to today's video lecture on the comprehensive approach to tackling viral diseases. In recent years, viral outbreaks have posed significant challenges to global public health, emphasizing the critical need for effective strategies in surveillance, infection prevention, control, and clinical care. This lecture aims to provide an insightful overview of the multifaceted methods utilized in combating viral diseases from initial surveillance to clinical management. Viruses are diverse and ubiquitous, causing a spectrum of illness. Climate change, population movements, and healthcare disparities create complex dynamics that must be considered when tackling viral threats. Mapping viral diseases involves the spatial representation and analysis of disease occurrence, spread, and patterns. Now, let us look into the modes of transmission of human viral diseases. Virus transmission may be horizontal or vertical. However, most transmission is horizontal, that is, between individuals within the population at risk. The route of entry is a major factor in determining the patterns of occurrence, mode of spread, and risk populations for each virus infection. Shedding of virus usually occurs from one of the body openings or surfaces also involved in the entry of viruses. With localized infections, the same body openings are involved in both entry and exit. In generalized infections, a greater variety of modes of shedding is recognized, and some viruses are shed from multiple sites. For example, hepatitis B virus, HIV, and cytomegalovirus in semen, cervical secretions, milk, and saliva. The amount of virus shed in an excretion or secretion is important in relation to transmission. Very low concentrations may be irrelevant unless very large volumes of infected material are transferred. On the other hand, some viruses occur in such high concentrations that a minute quantity of material, for example, less than 1 microliter, can transmit infection. Viral transmission by respiratory and oropharyngeal route. Many different viruses cause localized disease of the respiratory tract, are shed as aerosols in the mucus or saliva, expelled from respiratory tract during coughing, sneezing, and talking. Viruses are also shed from the respiratory tract in several systemic infections such as in measles, chickenpox, and rubella. A few viruses, for example, the herpes virus, cytomegalovirus, and Epstein-Barr virus, are shed into the oral cavity, often from infected salivary glands or from the lung or nasal mucosa, and are transmitted by salivary exchange through kissing or other social activities. Aerosols are most infectious in early respiratory infection at a peak of viral replication. But there is also a variation from individual to individual, and some patients, for reasons not well understood, seem to be more infectious than others and are referred to as super spreaders. Spread also occurs when fomites, including tissues, ward equipment, and household objects become contaminated with respiratory secretions or aerosol droplets, and contacts transfer this material to their own respiratory tracts. This mode of spread particularly involves those in close contact with others under conditions of poor hygiene. Environmental sources of airborne transmission include virus contaminated dust, thought to be the source of arenavirus infections, and aerosols of infected urine from rodents or bats. However, most respiratory viruses being enveloped are relatively labile and do not survive long outside the body unless kept moist in secretions. Spread through gastrointestinal route or the fecal oral route. Enteric viruses are shed in feces or vomit and the more voluminous the fluid output, the greater is the environmental contamination caused. These viruses tend to be hardier and are able to survive environmental conditions longer outside the body than the enveloped respiratory viruses. Two epidemiological patterns are seen. A point source outbreak occurs when many people may ingest contaminated food or water, for example, at weddings or other functions. This particularly occurs with salads, uncooked shellfish, or through drinking unsafe river water or well water contaminated with sewage. And two, person to person spread by fecal oral route is more gradual and occurs more efficiently in households without running water, hand washing facilities, or toilets where there is poverty and lack of education. Cutaneous route. The unbroken skin normally presents an impermeable barrier to virus entry. However, minor skin abrasions can be an important source of virus for diseases in which transmission is by direct contact, for example, warts. Several pox viruses may spread from animals to humans and sometimes from humans to animals through contact with skin lesions. For example, the viruses of cowpox, vaccinia, ORF, and pseudocowpox. Genitourinary route. Many viruses can be found in semen or vaginal secretions. Sexual transmission of virus infections by mucosal contact can be efficient because the virus is kept moist and does not need to survive long outside the body. However, dissemination through risk population by this route is usually slower than by respiratory spread because involvement of multiple contacts is usually slower. Blood-borne route. Just as viremia is an important source of virus dissemination with an individual host, so it is between hosts. Blood-borne transmission of viruses is more a problem among intravenous drug users due to contaminated needles and injecting paraphernalia. Of course, blood is usual source from which arboviruses are acquired by the arthropod vectors during the course of taking a blood meal. Less commonly, some arthropods, horseflies, other biting flies transmit viruses passively by contamination of their mouthparts and interrupted blood feeding on multiple hosts. Other routes of viral infection could be the ophthalmic route, wherein viruses can be introduced into the eye from the patient's fingers, from swimming pools, from inadequately sterilized ophthalmic equipments. Milk-borne route. Several viruses, for example, the cytomegalovirus HIV-1 and HTLV-1 are excreted in milk, which may serve as a route of transmission to the newborn infant. Transmission of virus from mother to embryo, fetus, or newborn is an important instance of cross-generational transmission that facilitates survival of some viruses in nature. This mode of transmission, referred to as the vertical transmission of a virus, may be lethal to the fetus and cause abortion. It may be associated with congenital disease or congenital abnormalities, or it may cause a subclinical infection. Human arthropod infections. The arthropod vector acquires virus by feeding on blood of a viremic animal or person. The ingested virus replicates initially in the arthropod gut and then in the salivary gland over several days. This period varies between different viruses and is influenced by ambient temperature. Virions in the salivary secretions of the vector are injected into new vertebrate hosts when the arthropod feeds on the new host. Arthropod transmission paves the way for the virus to cross species barrier, since the arthropod may bite birds, reptiles, and mammals that rarely or never come into close contact with each other. Vertebrate reservoir hosts are usually wild mammals or birds which generally sustain subclinical infections, producing an ongoing vertebrate arthropod cycle. Humans are rarely involved in this type of maintenance cycle called the enzootic cycle unless they venture into a site where the contact occurs with the infected vectors. Here, the spread of infection from a primary maintenance cycle in the wild to a cycle involving an amplifying host like a domestic animal species or a different arthropod species, and finally it comes back to humans. The regular reintroduction of infection from a non-human reservoir in case of zoonosis both assists in the perpetuation of a virus in human population and governs the distribution and extent of these infections. Example, the rabies virus. The extent of human infection depends on the degree of contact with the animal reservoir and prevalence of infection in the reservoir. The existence and possible extent of an animal reservoir is of fundamental importance in considering plans for regional elimination or global eradication of any human viral disease. Why is mapping viral diseases so important? Mapping viral diseases helps in controlling outbreaks. It helps in identifying hotspots, tracking transmission patterns, and guiding interventions. It helps in resource allocation. It helps in directing resources such as healthcare personnel and testing kits to areas with highest disease burden. It helps in understanding risk factors, in identifying environmental, social, and behavioral factors that contribute to viral spread. It helps in predicting future outbreaks, utilizing models to forecast potential outbreaks and to prepare accordingly. Data collection for mapping viral diseases. What are the important data required for mapping viral diseases? The confirmed cases. This is the primary data point used for mapping, representing individuals who have tested positive for the virus through diagnostic tests. Hospitalizations. Data on hospital admissions due to the viral disease can provide insights into the severity of cases and any potential strain on healthcare systems. Deaths. While not ideal for real-time mapping due to reporting delays, mortality data can offer a broader picture of the outbreak's impact. Travel data. Tracking travel patterns can help identify potential routes of transmission and inform public health interventions like travel restrictions. Demographic data. Overlaying case data with demographic information, for example, age, location, can help identify high-risk populations and tailor interventions accordingly. Environmental data. By analyzing the spatial correlation between environmental data and case distribution, areas with specific environmental conditions conducive to viral spread can be identified. Now let us look into the core mapping methods. Geographic Information Systems (GIS) is a spatial system that creates, manages, analyzes, and maps all types of data. This allows researchers to visualize the geographic distribution of viral cases on maps, uncovering patterns and identifying hotspots of transmission. This helps public health officials target interventions and resource allocation to high-risk areas. Epidemiological studies. Viral disease epidemiology is a study of determinants, dynamics, and distribution of viral diseases in populations. Epidemiology is effective in clarifying the role of particular virus and viral variants as the cause of disease, clarifying the interaction of viruses with environmental determinants of disease. determining factors affecting host susceptibility, unraveling modes of transmission, and field testing of vaccines and antiviral drugs. Now, let us look into some terminologies associated with epidemiological studies. The comparison of disease experience between populations is expressed in the form of rates. The terms incidence rate and prevalence rate are used to describe quantitatively the frequency of occurrence of infection or disease in populations. Incidence rate is defined as the ratio of new cases occurring in a population to the size of the population during a specified period of time. Prevalence rate is the ratio of the total number of cases occurring in a population to the size of the population during a specified period of time. Seroprevalence rate relates to the occurrence of antibody to a particular virus in a population. The term case fatality rate is used to indicate the percentage of subjects with a particular disease that die from that disease. An infectious disease is characterized as endemic when there are multiple or continuous chains of transmission resulting in continuous occurrence of disease in a population over a period of time. Epidemics are peaks in disease incidence that exceed the endemic baseline or expected rate of disease. A pandemic is a worldwide epidemic. Types of epidemiological studies. There are three basic analytical techniques used to investigate relationship between cause and effect and to evaluate risk factors of a disease. First, the case control study. A case control study is initiated after the disease has occurred. The purpose being to identify the cause by comparing the cases and controls. It is thus a retrospective study, that is, going back in time to determine the causative events. It requires careful selection of the control group matched to the test group so as to avoid bias, together with the careful selection of the questions and tests used. It can be used, for example, to answer whether a particular enteric virus can cause disease by comparing the rate of excretion of the virus in a cohort of children hospitalized with gastroenteritis with a group of age-matched children hospitalized for some other reason. Second, the cross-sectional study. A cross-sectional study is used to measure the prevalence of a given marker in a population and can be carried out relatively quickly. One caveat is that populations are often not homogeneous. A small subgroup at high risk may contribute most of the cases, and a major determinant of prevalence in any one study may be the size of such a subgroup in the total sample. For instance, blood donors have long been carefully selected group where individuals with risk factors for blood-borne diseases are excluded, and depending on the thoroughness with which this exclusion is done, blood donors may not be representative of the general population. Age-specific prevalence rates go further and give considerable information about historical changes and mechanisms of virus transmission. Third, cohort studies, usually carried out prospectively, start with a presumed cause or possible future risk. For example, a new treatment or vaccine. A population exposed to infection is followed over time in order to identify significant correlates such as the outcome of disease, reinfection, vaccine efficacy, etc. This type of study requires the recording of new data and the careful selection of a control group that is as similar as possible to the exposed group, except for the absence of contact with presumed causative influence or treatment. Such studies do not lend themselves to rapid conclusions as Groups must be followed until the disease is observed. Such studies are invariably expensive. However, successful cohort studies can provide strong evidence for cause-effect relationships and are essential for defining the safety of new vaccines. Long-term studies of families or larger groups, for example, the population of a city, can yield much useful information about the natural history of diseases and the long-term effects of, for example, interventions, chronic infections, or environmental factors. But such studies are expensive and require long-term dedication of both personnel and resources. Seroepidemiological studies, also known as seroprevalence studies, are a type of observational research used in epidemiology to understand the distribution and patterns of disease in a population by measuring antibodies in blood. Sentinel studies play a crucial role in virus mapping efforts by acting as an early warning system and providing real-time data on the circulation and characteristic of viruses in specific populations or geographic regions. For example, sentinel chicken flocks are set out for the early detection of presence of arboviruses such as St. Louis encephalitis virus in the southern and western United States. These flocks are bled and tested weekly for the presence of virus or antiviral antibody. They provide an early warning of the levels of virus amplification that occur before epidemics. Molecular epidemiology is the integration of epidemiology, the study of distribution and risk factors of disease, and molecular biology, the study of biological diversity and processes at the molecular level. The techniques used in molecular epidemiology include viral genome sequencing. Viral genome sequencing determines the complete genetic makeup of a virus. This process provides crucial information about the viral genome structure, organization, and genetic content. High throughput technologies such as the next generation sequencing technologies have revolutionized the field by enabling rapid and cost effective sequencing of viral genomes. Through viral genome sequencing, researchers can identify genetic variations, mutations, and potential virulence factors that influence the virus's behavior and interaction with the host. Phylogenetic analysis helps in reconstructing evolutionary relationships between different viral strains. By comparing the genetic sequences of viruses, researchers can build phylogenetic trees that illustrate their evolutionary history and divergence. It also helps trace the origins of viral outbreaks, track the spread of infections, understand the mechanisms during viral evolution. This information is invaluable for disease surveillance, prevalence, prevention, and control. Molecular dating estimates the time at which a particular virus emerged or diverged from a common ancestor. By analyzing the genetic differences between viral strains and incorporating molecular clock models, researchers can infer the timing of key evolutionary events. This provides insights into the emergence of new viral pathogens, the dynamics of viral evolution over time, and the factors shaping viral diversity. Understanding the temporal patterns of viral evolution is essential for predicting future outbreaks and devising effective intervention strategies. Genetic diversity analysis measures the extent of variation within a viral population. By analyzing the sequence data from multiple viral isolates, researchers can assess the genetic diversity, distribution of genetic variants, population structure of several viruses. Genetic diversity analysis reveals the adaptive potential of viruses, their ability to evade host immunity, and the emergence of drug-resistant strains. This information helps for development of vaccines, antiviral therapies, and diagnostic tests tailored to specific viral variants. Now, let us see the various ways in which the viral mapping data can be represented. The data visualization techniques. Choropleth maps. Choropleth originates from combining two Greek words, choros, which signifies region, and plethos, which means multitude. This type of map displays various data in different regions to visualize geographic data on a region by region basis. These maps represent data using different colors or shading patterns to indicate the variation in specific variables across geographic areas such as countries, states, provinces, counties, or other administrative divisions. Here is a choropleth map with labeled district names, number of confirmed COVID cases, and nearest testing centers in Maharashtra, India. Dot mapping is a cartographic representation method to visualize discrete absolute values and their spatial distribution. To achieve this, dots equal in size and represented value are used. According to dot value, a certain number of dots are used to depict a data value. These dots usually form dot clusters. The data value needs to be rounded to multiple of the dot value. It is possible to roughly determine the visual data value by counting the dots and multiplying this number with the dot value. Here is a dot map released by the World Health Organization depicting the distribution of COVID-19 cases as of 19th March 2020. Here, the dot value, the smallest dot value is 1 to 10 and the largest value represents more than 30,000 cases. Heat maps is yet another visualization technique which is used to represent spatial data by assigning colors to geographic areas based on the intensity of a particular variable. Heat maps effectively illustrate the geographical distribution of confirmed cases, allowing for quick identification of hotspots and potential transmission corridors. Monitor changes in case numbers over time and to reveal areas with escalating outbreaks or declining trends. Here is a COVID-19 heat map of the number of COVID cases in India. Cases less than 25,000 are represented with in green. Cases between 25,000 to 1 lakh are represented in yellow. Cases ranging from 1 lakh to 4,99,999 are represented with a darker shade of yellow, and cases more than 5 lakh are represented in red. Kernel density maps are statistical methods used to visualize the spatial distribution of virus cases by creating smooth continuous surfaces from point data. The purpose is to visualize the intensity and spatial patterns of virus occurrences, helping to identify hotspots and areas of high transmission. This is a kernel density map representing the geographic distribution of COVID-19 deaths across India. Deaths below 20 are represented in blue. Deaths of 1,700 and above are represented in red. Now, let us look into surveillance for newly emerging viruses. What is surveillance? Surveillance is an ongoing and systematic collection of routine data which are then analyzed, interpreted, and acted upon. The word ongoing in the description of surveillance helps to distinguish it from a survey, which is usually finite, tends to focus more on one or more groups of persons and involves a questionnaire. Nevertheless, surveillance does not continue forever. When it is no longer useful, it should be stopped. Essential ingredients of surveillance. To be systematic and consistent are the important ingredients of surveillance. If reporting centers are not consistent in what they report, nor regular, the data they send will be uninterpretable and probably useless. Defining what needs to be reported, its frequency, and agreeing on the criteria for making a reportable diagnosis are necessary to make sense of the data. Timeliness is another important ingredient of a surveillance system. This is especially the case with infection. It is particularly essential for the early recognition of outbreaks, but it is important also in evaluating the success or otherwise of programs to control the spread of an infection. Example, the effect of lockdown on coronavirus disease on a population. Representativeness is another important ingredient of surveillance program. The program has to be representative of the effect of that infection on a population. This may not always be possible, and any biases and inconsistency in reporting has been taken into account in interpreting the data. Completeness is generally less important than representativeness, especially for relatively common infections. Completeness becomes important if or when the number of infections decreases, whether on its own or due to a public health intervention. For some serious infections, completeness may also become important. The collection of routine data is another important characteristic of surveillance, especially with surveillance of laboratory infections. Generally, testing of samples is done for diagnosis, not primarily for surveillance. As laboratory testing is expensive, making further use of the results by contributing to surveillance makes much more efficient use of information, and the contribution to surveillance itself can often justify further testing. Now, let us look into the data collection methods for surveillance of viruses. Death certification. In most developed and middle income countries, deaths are certified primarily for legal reasons but have proved to be an important data source to use for surveillance. Clearly, they tend to be useful mainly for serious infections and may suffer from inaccuracies, but remain a useful basic data source. Diseases with a high mortality rate and a short duration of illness, the viral hemorrhagic fevers, for example, will obviously be better represented by death certification than those with a low mortality. It is well known that death certification standards of accuracy vary not only from country to country but also within countries. In England, in the early years of HIV epidemic, it was shown that deaths in single men had risen in number, although AIDS was not mentioned on the death certificates. With influenza outbreaks, there is usually an increase in the total mortality in elderly, otherwise unaccounted for. The reporting of deaths from SARS-CoV-2, both within and between countries, is also known to be inconsistent. Excessive deaths have become a useful measure in estimating mortality from influenza and SARS-CoV-2. Laboratory reporting systems can sometimes be another useful source of information on mortality from infection. Notifications. Surveillance systems such as statutory notification tend to be based mainly on clinical features. These can be very useful for common diseases with distinctive clinical syndromes such as measles and mumps. It is important, however, not to take a disease notifiable unless there is a good reason for it. Good reasons include a mass vaccination program when surveillance is virtually mandatory, any other mass control program, or serious diseases for which contact tracing, mass or close contact prophylaxis, or investigation into source is necessary. Seriously less common viral infections such as poliomyelitis are notifiable in most countries. This is because contact tracing and preventive measures can be taken. Broader clinical diagnosis such as aseptic viral meningitis may on the face of it be useful to notify, as it is usually impossible at the bedside to distinguish such cause of it. General practitioner surveillance systems are useful in providing data of epidemiological value for common infections that are not notifiable, such as common cold or chickenpox. They are, of course, clinically based, but nevertheless useful and often surprisingly accurate, possibly because general practitioners who subscribe to a surveillance system are motivated to do so. General practitioner surveillance systems also tend to be good for timeliness and completeness. Outbreaks of influenza are often first recognized by general practitioners. Laboratory data. It is useful to think of laboratory data as of being qualitative rather than of quantitative value, as they add quality and detail to disease surveillance. In the example of aseptic meningitis, a precise diagnosis of mumps, ecovirus, or Coxsackie virus is particularly useful in clustering and outbreaks. Indeed, as the enteroviruses exhibit a strong late summer or autumn seasonal pattern, with each enterovirus returning to its baseline in winter, when number of particular type continues to be reported at a level higher than the winter baseline, the return of this virus to cause another pandemic in the following summer can usually be safely predicted. Laboratory data are also essential in qualifying food poisoning and gastroenteritis. Separating viral from bacterial causes is a useful first step, as their management tends to be very different. Norovirus gastroenteritis can be foodborne, but also spreads very easily from person to person because an extremely small dose is necessary for infection to occur, and the virus being fairly resistant to environment will survive for some time. Management therefore must concentrate on hygiene. Rotavirus, on the other hand, is not commonly known to be foodborne. It can be distinguished from norovirus by its strong winter pattern in the temperate climates and in its preponderance in infants. With most bacterial causes of food poisoning, especially the salmonellas, management often depends on removal of the offending food. This is less common with the viruses. Only occasionally do hepatitis A or norovirus cause outbreaks of food poisoning. Laboratory surveillance is particularly essential for unraveling the mass of respiratory viral infections that inflict humans, such as respiratory syncytial virus, adenoviruses, parainfluenza viruses, and rhinoviruses. Indeed, it is particularly useful for influenza, not only separating it from influenza-like illness, but also in identifying influenza A and B, and if A, the subtype and variant. The constituents of every influenza vaccine depend on laboratory data. The process of surveillance. All approaches to surveillance share some common principles. At a fundamental level, surveillance aims to identify individual cases by using certain algorithms. Second, detect population patterns in identified cases and use pattern detection algorithm to identify patterns in them. Third, to report these patterns and to convey the information to decision makers about the population health pattern. Critical points in this process include detection of events in individuals, example diagnosis of measles, the identification of patterns in populations, example rise in incidence in a geographic location, and incorporation of these information about the identified patterns into decisions about interventions. Types of surveillance systems. Surveillance of outbreaks can be revealing and important to allow public health measures. Routine surveillance helps to monitor reported cases for trends and potential outbreaks. For example, in one incident, routine surveillance revealed 2 cases of hepatitis B associated with one tattooist. Inspection of notification data on hepatitis revealed 33 cases in all associated with the tattooist. Hospital admissions. These can be useful for certain more serious infections such as hepatitis and encephalitis. They can be unwieldy, generally lack detail, and often are published a year or more after the events have occurred. Serological surveillance has become increasingly important and is useful tool in assessing the immunity of a population. though it can also be used to identify vulnerable individuals. The immunity of a population can be vaccine-induced, and serological surveillance is a valuable adjunct to the methods available to monitor a mass immunization program. Vulnerable age groups can be identified and booster doses of the vaccine introduced. Surveillance of viruses in non-human and environmental sources. To build a picture of an infection, animals, birds, and the environment have been placed under surveillance. Rabies in foxes and other wildlife, influenza in birds, pigs, and other animals are examples of important and fairly successful surveillance systems. With the more recent examples of the emerging viruses such as Nipah and the Group B coronaviruses, this type of surveillance is becoming important. There are some surveillance systems for influenza A virus in avians and humans. Environmental surveillance of sewage and other wastewaters for wild and vaccine poliovirus as well as other viruses also exist. Newer types of surveillance systems. Syndromic surveillance. Syndromic surveillance complements traditional public health surveillance by collecting and analyzing health indicators in real time. It is usually based on an increase in an unusual group of clinical features which may lead to investigation. For example, the beginning of the outbreak of SARS-CoV-2 was an increase in an association of unusual clinical features in Wuhan, in effect a new syndrome. Although there was some delay in reporting this particular unexpected increase, the primary objective of this type of surveillance is to recognize an outbreak as early as possible. Sudden changes in the number of doctors' prescriptions for specific drugs, school or workplace absenteeism may also be used to uncover new outbreaks or new infections. Electronic surveillance systems are now being developed to detect new outbreaks. The fear of terrorism has stimulated this greater interest in syndromic surveillance. Syndromic surveillance will undoubtedly become important with time. Sentinel surveillance can be set up using a selected and willing number of motivated laboratories. Chosen laboratories would be expected to diagnose a reasonable number of cases and infection and should be within a fairly large area in which they would be expected to diagnose all cases of the infection and of course to be technically excellent. Sentinel surveillance systems tend to be time limited and are unsuitable for rare infections. Enhanced surveillance is a term used for infections of public health importance to combine epidemiological and microbiological data as well as other types of information necessary. It may include data from a range of sources. For COVID-19, clinical and microbiological as well as social data such as ethnic background have been collected by several countries including from asymptomatic persons. Enhanced surveillance is expensive on costs and manpower and should only be put into practice for special reasons such as pandemic. Active surveillance. This is a form of enhanced surveillance to ensure completeness and consistency. Reporting sources have to report, make negative returns whether or not they had cases. Tailored systems adapt to specific diseases. For example, the association of HIV/AIDS with stigma makes surveillance of this infection difficult. In the UK and some other countries with data protection acts, HIV infection, as with other sexually transmitted infections, is not notifiable. Special confidential surveillance systems through clinicians as well as laboratories are in place. Now let us look into infection prevention, control and clinical care. Measures to minimize transmission. Quarantine. Quarantine is used to separate and restrict the movement of well persons who may have been exposed to a communicable disease to see if they become ill. Quarantine is a measure first used by the Venetian Republic in the 14th century for the control of plague, literally meaning 40 days, quaranta giorni. The quarantine of shipping was also used by English colonists in North America in 1647 to try to prevent the entry of yellow fever, and smallpox. Quarantine proved very effective in keeping Australia free of endemic smallpox and in delaying the entry of pandemic influenza into that country in 1990. However, with the onset of air travel and the consequent arrival of passengers before the end of most incubation periods, quarantine as a sole measure of control has become less effective. Isolation is used to separate ill persons who have a communicable disease from those who are healthy. The isolation of individuals, patients suspected of presenting an infection risk is a routine and essential practice within hospitals. The measures adopted, for example, enteric or blood precautions and the stringency of isolation are governed by the characteristics of the virus involved. Sometimes, for more highly contagious diseases, more stringent precautions, special training, and personal protective equipment may be required. Measures to reduce the enteric spread of viruses. Hygiene and sanitation have a profound effect on the incidence of enteric infections. Viruses that infect the intestinal tract are shed in feces, and in many human communities, the recycling of fecal material back into humans through the oral route following fecal contamination of food or water is common. Hands contaminated at the time of defecation and inadequately washed may transfer viruses directly or indirectly to food, a particular problem among those responsible for the preparation of meals to be eaten by others. Education about adequate hand washing and provision of running water for toilet purposes are vitally important measures. In many densely populated parts of the world, there are no reticulated sewerage systems, and sewage may seep into wells, streams, or other drinking water supplies, particularly after heavy rain. Explosive outbreaks of many different gastroenteritis virus occur from time to time when sewerage mains burst or overflow and contaminate drinking water supplies. Raw sewage contains 10 to the power of 3 to 10 to the power of 6 infectious virus particles per liter, mostly enteroviruses, caliciviruses, adenoviruses, and rotaviruses. Titers drop 100-fold, typically 10 to 100 plaque forming units per liter following treatment in modern activated sludge plants because virions adsorb to solid waste and this sediments as sludge. The primary sludge is generally subject to anaerobic digestion, leading to a significant reduction of virus titer. Some countries require that treated sludge be inactivated by pasteurization prior to being discharged into rivers and lakes or prior to use as landfill or fertilizer in agriculture. In countries where wastewater is recycled for drinking and other domestic purposes, the treated effluent is further treated by coagulation with alum or ferric chloride, adsorption with activated carbon, and finally chlorination. Evidence that byproducts of chlorination are toxic to fish and may be carcinogenic for humans has encouraged several countries to return instead to ozonation. Ozone is a very effective oxidative disinfectant for viruses as well as bacteria, provided that most of the organic matter containing adsorbed viruses is first removed. Viruses also degrade over time naturally in the environment as they are exposed to heat, desiccation, and UV light. Also, storing water for a long time in lagoons results in virus inactivation. Measures to reduce the respiratory spread of viruses. Increased understanding about how respiratory viruses are transmitted via both aerosol droplets and contaminated fomites has led to improved guidelines to reduce respiratory virus transmission both between hospital patients and staff in the community. These guidelines include advice about hand washing after contacting respiratory secretions, care and disposal of used tissues, and advice to minimize transfer of contaminated objects, including stethoscopes, between individuals. Commercially available face masks have varying grades of performance specifications with respect to fluid resistance, breathability, and efficiency in filtering microparticles. Masks should be chosen appropriate to the degree of risk, the purpose required, and closeness of fit. Attempts to achieve air sanitation by filtration and or ultraviolet ray radiation in public buildings have proved to have only a marginal effect, although these measures are an important feature of biosafety cabinets widely used in virus laboratories. Respiratory viral infections remain very common, and congregation of large numbers of people in crowded public places and the frequency of air travel enhance the rapid spread of respiratory viruses to non-immune populations. This is seen most clearly with the newly emerging infections such as SARS and the 2009 H1N1 pandemic influenza. The population of the world now constitutes a single ecosystem for human respiratory viruses, although seasonal and climatic differences between the northern and southern hemispheres affect the incidence of particular diseases like influenza at any particular time. Measures to reduce the spread of sexually transmitted infections. Increased understanding of sexually transmitted diseases led to improved public health efforts. Public health campaigns focus on safe sex and routine sexual transmitted infection testing. Advances in treatment are prevention, post and pre-exposure prophylaxis. However, there is a complex interplay of various factors that affects outcomes. The global challenge being effective application of advances across diverse populations. Principles of safer sex. Seriously consider whether you want to have sex. Get tested. Be sure you know your and your partner's HIV status. Prevent exposure to blood, semen, vaginal, and other bodily fluids. Cover up body parts that could be infectious. Always use a condom and use a new one every time. Be in a monogamous relationship or limit your number of sexual partners. Get screened for other sexually transmitted diseases. If you or your partner have or has contracted a sexually transmitted disease, receive treatment immediately and do not have sex until your doctor says it's okay. Don't abuse alcohol or drugs which are linked to sexual risk taking. Talk openly with your partner. Remember, these discussions are easier with your clothes on. Transmission of blood-borne infections occurs through transfusion of infected, unscreened blood or blood products and organs, use or reuse of unsterilized medical equipment, needle stick injury to healthcare or laboratory workers, sharing injecting equipment by intravenous drug abusers, tattooing, body piercing, acupuncture, initiation rites, contact spot with bleeding. The control measures to prevent transmission. Screen blood and blood products for any potential viruses. Implement strict guidelines for medical procedures and equipment sterilization. Enforce single use of needles and equipment. Manage needle stick injuries with protocols. Regulate and provide guidelines for tattooists, body piercers, and acupuncturists. Offer harm reduction options for intravenous drug abusers. needle exchange, education, and rehabilitation. Now, let us look into the measures to reduce the spread of viruses by using the vector control method. Because the control of non-human vertebrate reservoir hosts is usually difficult to achieve, control of arboviruses may be approached by directly reducing the number of arthropod vectors and by minimizing human exposure to these vectors. The philosophy of an integrated pest management is widely used. It consists of a combination of approaches selected for a particular problem and with the concern for the full environmental, health, and economic context. The targeting of vectors includes the elimination of breeding sites and direct destruction of adult mosquitoes or larvae. The flight range of many vector mosquitoes is so limited that much can be achieved by concentrating on the immediate vicinity of human settlements, particularly in case of species attracted to human habitation such as Aedes aegypti. Any still water constitutes a potential breeding site. Swamps and ditches should be drained and water collecting refuse such as discarded tires, tins, cans, and plastic containers be destroyed. Domestic pot plants, wet shower flows, toilet water tanks, and rainwater tanks pose special problems. Larvicidal chemicals can be placed in domestic jars and kerosene or diesel oil layered on the surface of non-potable water. The biological control of mosquito larvae is also an alternative, albeit less developed, control strategy. Approaches include the introduction of fish species that ingest mosquito larvae and introduction of sterilized male mosquitoes in order to reduce insect breeding. The use of Bacillus thuringiensis, which releases crystal toxic to mosquito larvae, has been also attempted. The introduction of bacterium Wolbachia has been found to reduce the capacity of Aedes aegypti mosquitoes to transmit dengue, chikungunya, and yellow fever viruses. However, the growth of cities in developing countries, largely by the growth of urban slums, has greatly accentuated the difficulties of control of domesticated Aedes aegypti mosquitoes. The Asian tiger mosquito, Aedes albopictus, an aggressive daytime biting species, an important vector of dengue and chikungunya viruses, has spread widely since the 1950s and now covers large areas of the world. Once established, it is difficult to eradicate. For most arbovirus infections, other than urban dengue and yellow fever, the vectors breed over too wide a geographical area to make vector control feasible on a local scale, and then only in the face of a threatened epidemic. The use of insecticides is a controversial issue because there are frequent objections from environmentalists and mosquitoes eventually develop resistance. Any policy should be decided on the basis of risk-benefit analysis on a situation-by-situation basis. Some countries base their arbovirus control programs on aerial insecticide spreading, but most retain this approach only for emergency control in the event of an epidemic, specifically aimed at rapid reduction of adult female mosquito population. Organophosphorus insecticides such as malathion or phenitropothion are delivered as an ultra-low volume aerosol generated by spray machines mounted on backpacks, trucks, or low-lying aircraft. Spraying of the luggage bags and passenger cabins of aircraft with insecticides may reduce the chances of intercontinental transfer of exotic arthropods, whether infected or not. Avoidance of exposure to the bite of arthropods is the other mainstay of control. Personal protection against mosquito bites can be achieved by use of screens on doors and windows, nets over beds, long-sleeved protective clothing, especially at dark, and repellents. In developed countries, the major barrier has been the whole house air conditioning. Insect repellents containing the chemical N,N-diethyl metataulamide DEET are particularly effective and do not present a health concern when used as directed. One of the ways to prevent viral infections is immunization. Immunization is directed primarily at making the individual resistant to infection and in many cases reduces the incidence of new cases. It can substantially reduce the source of further spread. Immunization may be active immunization or passive immunization. Active immunization involves the induction of an immune response by administration of an antigen or vaccine. Although the principal aim of active immunization is to protect the vaccinated individual, vaccination on a sufficiently wide enough scale will also enhance herd immunity to such an extent that transmission can be restricted or even arrested altogether within a given community or country. Passive immunization. Passive immunization confers short-term protection through the intramuscular or intravenous inoculation of preformed antibody, either using immune serum or immune serum globulin. Human immunoglobulin has replaced animal-derived preparations because heterologous protein may provoke serum sickness or anaphylaxis. Passive immunization is used in specific situations such as emergencies during travel or to protect newborns. The vaccination programs should consider vaccine characteristics, disease characteristics, and community factors in order to be effective. Now let us look into some clinical care measures for viral infections. Antibiotics are ineffective against viral infections. Treatment of viral fever depends on the type of infection and severity of symptoms. Respiratory infections usually clear up on their own. Manage fever and headaches with mild pain relievers. Use a hot bath or shower for sinus congestion. Get plenty of rest and fluids. See a doctor for a high fever that lasts more than 3 days. Gastrointestinal viruses are untreatable but usually go away quickly within 1 or 2 days. Drink plenty of fluids and eat a balanced diet. See doctor for high fever over 38 degrees centigrade, bloody stool or vomit, or inability to keep fluids down. Wash hands regularly to prevent spread. Clinical care for exanthemus viral illnesses. A viral exantheme is an eruptive skin rash that is often related to a viral infection. Exanthemus viral illnesses may be treated with antiviral or fever-reducing medications or topical ointments. See a doctor for any bleeding as it can be serious. Neurological viral fever may be treated with over-the-counter medications for mild symptoms. Always seek urgent medical attention for infections like meningitis as they can be fatal if left untreated. These are a few of the references for further reading. Now let us discuss some multiple choice questions. Which of the following methods is commonly used for mapping the global distribution of viral diseases? A. Serological testing. B. Molecular diagnostic techniques. C. Geographic information systems. D. Antibiotic susceptibility testing. The correct answer is C. Geographic information systems. What is the primary purpose of surveillance in context of viral diseases? To track the spread of antibiotic-resistant bacteria, to monitor the effectiveness of viral medications, to detect outbreaks and trends of viral infections, or to promote the use of prophylactic vaccines. The answer is to detect outbreaks and trends of viral infections. In clinical care of viral infections, what is the primary goal of antiviral therapy? To eliminate bacterial pathogens from the body, to reduce the severity and duration of symptoms, to prevent viral transmission to others, to boost the immune system's response to infection. Answer is to reduce the severity and duration of symptoms. Which of the following measures is not typically included in infection prevention and control strategies for viral diseases? A. Routine vaccination programs. B. Hand hygiene practices. C. Antibiotic prophylaxis. D. Personal equipment like masks, gloves, etc. Answer is C. Antibiotic prophylaxis. Which of the following factors contributes to the global spread of viral diseases? Availability of clean water and sanitation, improved healthcare infrastructure, international travel and trade, strict border control issues. Answer is international travel and trade. What is the purpose of serological surveillance in monitoring viral diseases? A. To identify the genetic sequence of the virus, to detect the presence of antibodies in the population. C. To determine the effectiveness of antiviral medications. D. To measure the viral load in infected individuals. Answer is B. To detect the presence of antibodies in the population. Which of the following infection prevention measures is recommended during a viral outbreak in a community setting? A. Mandatory use of antibiotics for all individuals. B. Quarantine of asymptomatic individuals. C. Promotion of social distancing and mask wearing. D. Administration of antifungal medications. Correct answer is C. Promotion of social distancing and mask wearing. Thank you for your kind attention.